next speaker. And as I mentioned, in the previous presentation, we've actually seen some sort of new types of maps, new types of information that we would like to deliver to the public health authorities. And actually, the next presentation is really about how do we communicate better with the control programs? How do we give some new insights that we can get from our genetic surveillance? And how do we empower the experts in the countries to better analyze our data and to do their own investigations from the data we deliver to them? So I would like Areeya to tell us a bit more about this and about the training programs that she's working on. Thank you.
Okay, thank you very much, Professor Areeya, for the introduction. And first, I would like to add more for the audience. If you have any question, could you please type your question in the Q&A box, and moderator will pick up your question at the end of this session for the asking to the panelists. Okay, now I will start my presentation on the expanding genetic surveillance through the communication and training. So in this section, in this topic, I will give you more overview on the how can we conduct the genetic surveillance network in each country in GenRe-Mekong project, and especially in Laos, that we have experience to find the requirements and that there are the outbreaks in Laos. So how can we use this genetic surveillance network to communicate and to provide the information to the national control program and stakeholder? So first, okay. So this is the overview of our project that yesterday maybe Professor Areeya already gave you some overview of the project. So our project would like to promote the application of malaria parasite by using the genetic surveillance to support decision making in malaria control and elimination efforts for the national malaria control programs. So in our project, there are three groups of partners. First, we include the national malaria control programs that have the information about the malaria situation and also have the authority to make the drug policy for malaria control in each country. Second, we have the local and international scientific partners. And the third one, we are GenRe-Mekong Scientific Support to give the support on the technical and also coordinate with the ethical issue and also any process in each country. After we have the our project have the main objective that would like to use for the data from the genetic surveillance into main topics. First, for the control program, just like to see how the drug resistance occur in each country, how about the treatment efficacy and outbreaks intervention. The second one is about the genetic epidemiology, including the drug resistant markers, parasite strain, population structure of parasite in each country, and transmission intensity and the imported case, and just like the cross-border transmission of the parasite. So to get to this use case, there are four main actions that we do in our project. First, it involves with the local sample collection and processing in each country. Second is about the country-specific and collective data analysis and sharing the data between countries. The third one is know how to translate and interpret genetic data in public health context. And you know that maybe the genetic data is quite hard to understand by in general. So we try to use the way and make the content of the information from the genetic to give the interpretable or easily to understand to the public health. The last one, we have the activity to give the result back with the genetic report cards, or just like in case and try to make like a maps or country report that easy for the programmatic use, and also some of scientific report that we will publish later. So this is the activity in our project in the first phase of GenRe-Mekong, and now in the second phase is in the translational phase. So first we have to know what is the requirements from our stakeholder. To get the requirements, we have like a virtual meeting, and also we try to get to know what is the problem in each country from the questionnaire and also from the literature reviews. After we get the requirements, we have the operations and analysis and translation in many activities, just like in the technical training. In the first phase, we have like a few training for the staff to know how to collect the sample and data. And we plan to have the online training for the scientific partner and also selected National Malaria Control Program and other scientific partner that would like to engage more in data generation, analysis, and interpretation that we can use for the regional database. And for the effective way to communicate to the National Control Program after we get the information, we make the summary report for the policymaker, and also we provide some in future we think to have like a dashboard on maps that easily to see the outbreak or any drug resistant marker in this region. And like now we plan to have also online materials that you can know how to use the genetic data for epidemiology use. And just like now we have the knowledge exchange, that's like a scientific forum, and the way that people can exchange to know the data and also how to get the sharing result or project implementation experience that we can share among our stakeholders. So now we will go to see what we have did for the Laos, for the malaria epidemiology in Laos. This is the background for the malaria in Laos. So in Laos now they use artemether and lumefantrine is the first line drug to treat the uncomplicated confirmed case. And the point of concern is previously there are the pre-existing of artemisinin-resistant parasite, especially they have the problem with the spread of the DHA-PPQ resistant parasite across the subregion, okay, in the southern Laos, even though the people has not been used in Laos. And recently they have the outbreak in the Outbreak province. So from this point, we try to use the genetic surveillance of malaria parasite to providing affordable and monitoring of the parasite response to different treatment at medical centers with few resources. Okay. So from this genetic surveillance, we think we hope that we can make an early detection of resistance. So it will lead to timely action to prevent the spread of the drug or the outbreak of malaria or drug-resistant malaria and limit the public health consequence. And another one is for the outbreak characterization, as you can see from the previous presentation from Dr. Valencia. So now we go to the first phase. What we have need to make the network workable or capable in Laos. So there are three main members. So first is National Malaria Control Program. We include the Center of Mariology, Parasitology, and Entomology, or CEMPE, from the Ministry of Public Health, because they have authority for the control and elimination policy. And that is the good thing that in Laos, Ministry of Health classify our project or the GenRe-Mekong project as a surveillance activity. So for the national benefits, so we don't need the required additional informed consent as like for the as like the way for the do the research. So we can collect the sample in real time. Yes. And next, we have the scientific partner from Laos Oxford Hospital Wellcome Trial Research. So from the Lomru, they provide the well-trained staff to have the local sampling network, get the dry blood sample collection, and also data management. So we have the coordinator, and also they provide the protocols to each site clinical site in our field to get the sample correctly. And after the sample have processes and got from the site, it's sent to the GenRe scientific support and coordination. So we have the standardized protocol for sample collection, processing, and data analysis, and also to support the effective use of the data. So in our project, we try to develop data analysis pipelines and tools for translate genotype data into more interpretable and actionable forms. So the thing that we hope to see is data sharing in this sub-regional, so we can make like a overview and try to use the data to sharing and to solve the problem at the sub-regional level. Okay, so this is from the phase 1. We, as Dr. Gao mentioned that the malaria is foci of the that have the problem in the 5 provinces in southern Laos. Okay, in Savanakhet, Sarawak, Sekong, Japarsak, and Atapu. So in these 5 provinces, we have 107 study sites over there. There are the coordinator to go to the site and then do the sample collection, and also they have training field staff to get the dry blood spot from the figure. Okay, as you can see from the figure, and also they have the communication with the community to get more convenient to proceed the our project. So from the phase 1, we can have output and impact. That the one thing that we can see clearly. So from the genetic surveillance, the Ministry of Health in Laos used it to change the policy, because they saw that piperacillin resistant strain is spread across the border, and also in the southern Laos. They changed the policy, and then they used the artemether-lumefantrine for the first line. And the second one, so from this from the activity in the phase 1, we can attribute the effective genetic surveillance network in Laos with the well-trained staff, so we can promptly to go to the next step if there are something for the monitoring and detection of resistance. So we can see, so just the time that we translate from the phase 1 to phase 2, there are the notice, and from the same way that there are the increase in the falciparum case in other province. So we can proceed our project directly for the characterized outbreaks in the phase 2. So this is the way that we use our communication network, and also well-trained staff to do the things. First, there are the sample collection in the Atapu province. Promptly after we get the information that are the case number increased, and do the empirical sequencing. At the time, we did it in the Sangor. Okay, Wellcome Sangor Institute, and do the data analysis, so we can know about the drug-resistant mutation and prediction phenotype. And also we can see the network connection or genetic connectedness and population diversity of the parasite, as you see from previous data from Dr. Valanya. After we got this information, we have communicated back to Sempe directly. immediately after we got the result. So we report and discuss on the spread of drug-resistant parasite in Outbreak province. And after communication, we know we got the feedback from Sempe that they would like to know more about the MD1 copy number, because they consider that they're afraid that there will be the resistance to artemether or artemether lumefantrine that they use in the as a first line in the country. So for the extended network of communications, after we got the feedback from Sempe, we communicate to our group by Dr. Nien. She have the lab in our crew that can assay for the MTR1 copy number. And also we make the additional analysis to see the origins of the emerging of drug-resistant parasite. After we got some initial information, so we communicate back, because we think of the data usage and to prevent the spreading of drug resistance into sub-region. So we try to communicate back to the WHO also to have the response for this outbreak. So this is the summary that we have did along with the thing that we have in the outbreak. So from outbreak confirmed that we got the information for the sample, and we respond immediately for the collection of the sample by the Lomlu to describe the sample in Outbreak province, and we determine the cause of outbreak using our technology, that the empirical sequencing, and communicate back to all of the stakeholder to have the to try to see how we can control the outbreak with the sample and the WHO, and further to make it sustainable and to monitoring the prevalence of this strain. So now this is the challenge for us to have such a kind of the way to sustain the system to respond for the spreading of the malaria immediately. So first, we need to train and think of the available online platform for selecting, visualizing, and retrieving genetic epidemiology data. And also, data sharing is the one that is important, even international level or international level, to support regional surveillance. And another one is the planning and adapting a sustainable surveillance system to extend to new use cases that will be necessary in the final step toward elimination. So to make these challenges happen to success, we use another activity, many activities to cope with this challenge, including the in-country laboratories. Just now we have the central lab in Vietnam in our crew that will support the sample analysis in this sub-region. And another one is for the data analysis, we developed the GRC Malaria package that we use for processing data and analysis, create maps, and also to visualize population structure that easy to understand for the public health staff. So from this resource, in future, we would like to have the training course and meeting for the scientific partner and MCP and academics that we help them to make themselves to do the analysis by themselves and provide information to marine control program in timely manner. And also, as we have now, we have the scientific forum and also joint regional data event that we can share information together, and we know the situation of each country, so we can have a response immediately. Okay, so finally, I would like to thank to Sempe and all of the staff in field, okay, to collect the sample, and also Longlu for the scientific support, Moru and all crew that providing support for the laboratory, and also welcome Sanger Institute for the data analysis. Thank you very much.
Thank you very much, Areeya. This overview of how we work together with the control programs. And you know, the last couple of years, unfortunately, have been difficult in terms of communicating with the control programs. And what we hope is that the type of work that we've done with SIMPE, we will continuously be doing with the control programs in all the countries. So there was some talk about training. This is going to come live online in the second half of this year. And what we are hoping is that we're going to make it very accessible, such that different levels of expertise can be accommodated. You will not need to be a genetics expert from day one. And the idea is to really sort of help the development of local knowledge. For that purpose, I would actually call on all our country partners, you know, to highlight, if you know of other partners we can engage in these exercises. We would very much like to train, you know, university PhD students or technical members of the control programs and other people in the countries, so that we can form a sort of big community of people who work together to analyze the data and to share it and so on.